Hi, everyone. My name is Natalie O'Connor, a graduate certificate student in the Tropical Medicine program at the John A. Burns School of Medicine. And today I'll be presenting my project titled Detecting Pathogenic Leptospira along Manoa Stream. To begin, leptospirosis is a zoonotic bacterial disease transmitted through water or soil contaminated with urine from infected animals. It can cause a wide range of symptoms, from mild flu-like illness to severe disease, including kidney or liver failure. Hawaii has one of the highest U.S. incident rates, with several Oahu streams testing positive for Leptospira. Statewide analysis from 2012 to 2024 shows higher case rates in regions with increased rainfall and environmental exposure. In 2001, a notable outbreak occurred on the UH Manoa campus following a flooding event. The purpose of the study is to detect and quantify pathogenic Leptospira using the LIP-L32 gene in Manoa Stream and evaluate its spatial and temporal variation in relation to environmental patterns. For methods, we collected at least 1,000 milliliters of water from 5 sites along the stream on a weekly basis, spanning from October 2025 through January. These sites were chosen to follow the start and end of the watershed. Each sample was filtered through a 0.22 micrometer filter to capture bacterial cells, and DNA was extracted using the Qiagen DNeasy PowerWater Kit. We then performed quantitative PCR targeting the LIP-L32 gene, which is only found in pathogenic strains. Looking at the main results in Figure 1, each row represents a collection week, and each column represents one of the 5 sampling sites. Green dots indicate positive detection of the LIP-L32 gene, while black dots indicate negative results. A positive detection was defined as a CT value less than 40 in the qPCR assay. We also included negative controls, no template controls, and positive controls, all of which performed as expected, confirming the reliability of the assays. Across all weeks, we observed that every site had at least one positive sample, indicating that pathogenic Leptospira is present throughout the stream. However, detection was not uniform. The highest frequency of positive samples occurred at midstream sites, more densely populated areas, while the upstream and downstream sites showed lower detection frequencies. In Figure 3, we compare weekly positive detections with environmental data, specifically precipitation and temperature. What we observed is that increased detection of pathogenic Leptospira tends to follow periods of higher rainfall and slightly lower temperatures in the preceding week. For example, during weeks with higher precipitation, we often see a corresponding increase in positive detections. This suggests that rainfall may play a key role in mobilizing Leptospira into the water system, likely through runoff or environmental disturbance with an increase of vector presence. Overall, these findings highlight both spatial and temporal patterns of pathogenic Leptospira presence in Manoa Stream. Pathogenic Leptospira was widespread, detected in 29 out of 55 samples, or 52.7%, but more concentrated in midstream regions, and detection appeared to be influenced by environmental factors such as rainfall and temperature. In conclusion, this study demonstrates that Manoa Stream contains detectable levels of pathogenic Leptospira across multiple sites and time points, with increased prevalence associated with recent rainfall and cooler conditions. These findings are important for understanding environmental risk and can help inform public health awareness, especially for individuals engaging in recreational or occupational activities in freshwater systems. We would like to thank the Department of Tropical Medicine, as well as my mentors, Dr. Chang and Dr. Jourdan McMillan, for their guidance and support. We also thank the MHRT program for funding and training, the PATH group for their contributions, especially performing qPCR, and the SOAS group, including Dr. McMillan. I would also like to thank lab volunteers, Stephanie and Yugia, for all of their efforts in the lab. Thank you.